Once you have a question about decomposition in mind, how will you test it? How do scientists study decomposition? Studying decomposition can be tricky because it can take years for a leaf to decompose and decades to centuries for wood to decompose. It's hard to keep track of a leaf on the ground for that long. A very common way we study decomposition is using what are called litter bags. Essentially, scientists put the dead plant material, the litter, into a mesh envelope or bag. Commonly, the envelope is made out of a screen very similar to what is used in your windows at home. They often look like this. The mesh allows the small decomposers to get into the litter to do the decomposing, as well as water and soil, but keeps the litter contained in an envelope so that the scientists can pick it up to analyze how decomposition is progressing. By changing the size of holes in the mesh, we can change what types of organisms are allowed to reach the litter. By changing what litter we put into the bag, we can ask questions about how different plant species decompose differently. At the beginning of the experiment, we make a lot of litter bags. We put all of the litter bags outside on the ground. We decide how long we want to study decomposition, maybe a few months or a few years. Then, periodically, we collect a few of the litter bags at a time. For example, if you want to study decomposition over 3 months, you might collect a third of the bags each month so that you can see how decomposition happens over the entire period of time. So how do we answer our questions? For example, how might we measure the different rates of decomposition in the city versus the desert? One simple way is to measure how much mass has been lost from the bags at each location. How would we do this? We could standardize the amount of leaf litter in each bag at the beginning of our experiment. Then as we collect the leaf litter bags from our experimental sites, we would allow them to dry. Then we can re-weigh them and record how much leaf litter remains. The difference is decomposition. Students in a classroom can also record other important visual observations about decomposition, such as changes in color over time, how it loses thickness and becomes more fragile, and overall how sections of the leaf start to disappear. Sometimes you can see the fungi growing on the litter. You can also look at the amounts of tiny invertebrates that are eating those fungi and bacteria, if you make the right setup. Using light, heat, and household materials, you can separate the arthropod invertebrates from the soil. Since arthropods are generally ectothermic, they rely heavily on the surrounding environment to maintain appropriate body temperature. They prefer the cool, moist soil, so in these tall grain extractor funnels we use a light bulb to heat them up and drive them out of the soil where we can observe them. You can learn how to construct a tall grain funnel in an upcoming video. Decomposition can be due to organisms breaking down plant matter, also called biological decomposition, or it could also be caused by photodegradation from the sun, or abiotic decomposition. We can study photodegradation by using litter bags made from plastic that either allow or block UV radiation. By the way, in professional soil ecology labs, sophisticated instruments can assist us in determining how much of these decomposition processes has occurred. They can even analyze the relative amounts of remaining carbon, nitrogen, and phosphorus in our leaf litter samples. So what do you predict? Consider all you have learned about decomposition. Which conditions favor biological and abiotic breakdown of plant material in the soil? Where would it occur faster? Why? Remember how much fun it can be to explore in the soil? Now you are ready to ask your own questions about decomposition and conduct your own study. Happy digging!